up in Traverse City of all places. I'm feeling so sorry for her, Alex. Don't forget to sign up on the sheet here, and that keeps the pizzas flowing. Andrei Lomize is a senior research scientist in the Department of Medicinal Chemistry. That's an old term for a very important thing, and it's in our College of Pharmacy. Obviously, you know, the structure of molecules and how molecules interact with, small molecules interact with receptors and structures is so hugely important to understanding of how pharmaceutical agents work. And what we're going to hear about is really a number of very informative protein structural tools and capabilities that Andrei, you've developed over many years' time. And you know, it's really going to be a kind of a whirlwind high-level tour of not only your work, but it's going to be informative, you know, of what's done in pharmaceutical, in medicinal chemistry. And specifically what's interested in is looking at water-soluble and membrane-soluble proteins. The G protein-coupled receptor might be a good example of the latter. And so we're so grateful that you're here, and we're looking forward to learning about what you do.
Okay, thank you.
And about the field. Thanks for coming.
Thank you.
Yeah.
So today I would like to tell about development of computational tools and databases to study proteins and drugs in memory. And most of these tools and databases were developed in our group and during many years. And they have a number of unique capabilities not found in any other computational research. So biological membranes separate cells from the environment and create numerous intracellular compartments. And they are highly dynamic and have very complicated composition. The lipid bilayer itself includes more than 1,000 different species of lipids in eukaryotic cells. And it can be covered by cellular matrix, by glycocalyx, and from the intracellular side, it may interact with cytoskeleton. But the most important component of biological membranes are proteins. Actually, a human proteome includes more than 5,000 transmembrane proteins and even more peripheral proteins. The proteins can be transmembrane, which are integrated permanently in the lipid bilayer, and peripheral proteins, which interact with surface and can be only temporarily attached. So more than 5,000 transmembrane and peripheral proteins, and in general, more than half of all proteins in human organism interact with biological membranes at some time. And again, these membrane proteins form multiple interaction networks, participate in pathways, involved in all vital processes such as signaling, transport, metabolism, and so on. And it's also important to know that more than half of all pharmaceutical drugs target different membrane proteins, such as, for example, GPCR, classical example. So, what we would like to know about these proteins? We would like to know their three-dimensional structures. We would like to know how they are spatially arranged with respect to the lipid bilayer. And for peripheral proteins, we would like to know their membrane binding affinities. Also, there are many small molecules around which can penetrate through the lipid bilayer. And some of them are relatively hydrophobic. It could be drugs, could be metabolites, regulators, and so on. And others require transporters to be transported through the lipid bilayer because they are polar. So we are developing computational tools precisely to address some of these questions. Today I will tell about three different topics and three different types of computational modeling. First will be modeling of spatial positions of proteins in membranes and their hydrophobic thicknesses. This is our PPM program and web servers and OPM database. Another subject is modeling three-dimensional structures of relatively simple transmembrane alpha helical complexes, which are usually composed only one or two transmembrane alpha helices, like transmembrane gamma dimers and heterodimers. This is HealthMap and TenDoc, web servers and Membranome database. And the third part is modeling passive permeability of small molecules or cell-penetrating peptides through the lipid bilayer. This is Perm or CellPM web servers and our Perm database. So it will be 3 parts in my talk today. And altogether we created more than 5, we created 5 web servers and 3 databases. And actually, creating new computational methods is a difficult business. And there is also this equation, how reliable these methods are, how useful they are, and so on. So when we develop these tools, we use a number of criteria, general criteria. First, all our methods are physics-based and operate with different free energies, such as free energies of binding, folding, and transfer of different compounds, of proteins, peptides, and so on. And these quantities usually can be experimentally measured and compared with results of computation. Then we try to extensively test our methods against all available experimental data, and we also implement them as easy-to-use web tools, so everyone can use them or independently verify how they work. And we believe that if we develop something good, it can be applied on a very big scale. So it can be applied for entire proteomes, for modeling, for example, transmembrane gomodimers or single helices, or for entire protein data bank and so on. So we did wide-scale applications, and the results of our large-scale applications are available in our databases, PM and Perm, so everyone can look at them. So one of the important features here, probably the most important, that we are trying to reproduce different experimental free energy and contributions in our modeling based on that. For example, we have developed an isotropic solvent model of the lipid bilayer to calculate energy of transfer of small molecules of proteins from water to the lipid bilayer. We developed a thermodynamic model of alpha helix formation to reproduce thermodynamic stability of alpha helices when they are bound to micelles or membranes. And we developed a new method in force field to intermittent free energy of transmembrane alpha helix association, TMDOC program. And in addition, we don't use molecular dynamics usually here, and that's why our methods computationally are relatively fast compared to ND. And so there are many different transmembrane proteins in the PDB, for example. This is like part number 1, about spatial arrangement of proteins in membranes. And some of them have alpha helical architecture, and others have a beta barrel architecture. And there are also so-called bacterial pore-forming toxins, which may exist as water-soluble proteins just in water, but then they aggregate with each other, and they release hydrophobic parts of the structure, such as beta hairpins or hydrophobic alpha helices, and they form a channel in the membrane irreversibly, and this channel usually kills the cell. So there is a great variety of transmembrane proteins with different functions. But and these structures are available in the protein database. But we don't know how exactly these proteins are arranged with respect to the lipid bilayer. We don't know where there are so-called hydrocarbon boundaries, which separate lipid acyl chain region, the most hydrophobic part here, from the head group region of surrounding regions, where there is a significant amount of water and basically polar environment. And important parameter is the distance between these boundary planes, because it defines hydrophobic thickness of transmembrane proteins. And this hydrophobic thickness may be different for proteins from different types of biological membranes of different classes. For example, if you do these calculations, the beta barrels in outer bacterial membranes have relatively small hydrophobic thickness, around 24 to 25 angstrom, and they are thicker in proteins from the inner bacterial membranes, and even more thicker, a little bit like in average 33 angstrom in proteins from plasma membranes of eukaryotic cells. So this is an important parameter, hydrophobic thickness, which we also need to determine here. So what we did is relatively simple. We take the three-dimensional structure from the protein data bank. This could be peripheral or transmembrane protein. We position them in the lipid bilayer using our program, and we put results in our OPM database, which currently includes around 10,000 DB entries. But how we do it? We basically try to find the lowest energy arrangement of the protein in the lipid bilayer. To do it, we do a rigid body optimization of transfer energy of the macromolecule from water to the hydrophobic environment. And this transfer energy function includes first salvation energy term contributions, like hydrogen bonds between, for example, protein and water or lipid solvent, and electrostatic term related to dipole moments of the protein, and also contribution from ionizable groups. But in order to use these equations, these equations use some parameters like implicit salvation constants for different types of atoms, and to determine them, we need to know, we can determine using other equations, and to do that we need to know how polarity of the membrane change in different positions along the bilayer normal, across the lipid bilayer.
So you don't need like an enthalpy or a entropy term.
Basically, usually they are combined together, yes. For calculation of salvation energy, it is very difficult to separate enthalpy and entropy contribution. So it is empirically done for this sigma. Sigma, for example, includes hydrophobic interactions, which are of entropic nature, and also may include hydrogen bonds, which are of enthalpic nature. But the second term, to go back, this second term, electrostatic, this is purely enthalpic term. And this may include, actually, because it depends on pKa groups, so it may include actually both. But we're not trying to separate enthalpy and entropy completely. In another approach, which will be one of the next, we will use specifically conformational entropy of side chains, the changes in conformational entropy of side chains when two alpha helices are straight with each other. This is explicitly entropic contribution. So here we actually, when we tried to develop this method, we found that only 3 polarity parameters of the solvent will be sufficient. And these parameters are known like hydrogen bonding donor and acceptor capacity of the solvent, and dielectric constant, also called thermochromic dipolarity parameter. These parameters are widely used for organic solvents to describe their solubility properties. And that is exactly what we do here. We consider interior of lipid bilayer as a solvent, but as an anisotropic solvent. So the property, these polarity parameters can change along the bilayer normal. And in order to calculate how these parameters change, we use distribution of different lipid groups across the bilayer. It was like for UPC bilayer. And these distributions were experimentally determined and published using X-ray and neutron scattering for fluid bilayer. So based on this distribution, it is possible to determine how these parameters are changed along the normal, and then to calculate these parameters in different positions.
Well, you know, I hadn't really thought about this before, but you showed that really remarkable structure that looks like a tree almost, and some of it's in the membrane and some of it's outside.
Yeah.
And my guess is that, you know, the outside part, which has a different solvation energy, different set of terms, is as important, you know, a contributor in, you know, maintaining the whole structure as, you know, that part that's in the membrane.
Absolutely, yes.
And you want to model both of those.
Yes. So but here we don't model the three-dimensional structure, we only model position of well-known three-dimensional structure in the lipid bilayer, how it will be arranged to minimize this kind of free energy function. But this is actually an original approach which no one used before for lipid bilayers. That kind of implicit salvation model derived from experimental distributions of different groups. And we did it for 10 different model bilayers, not only UPC, and we also try to do it for even biological membranes using distributions of polar groups in the three-dimensional structures of transmembrane proteins. And we published something about it. So actually these distributions would be different from some kind of effective biological membrane. So, for example, using this approach, we can calculate spatial arrangement in membranes, not only transmembrane proteins, but also peripheral proteins. Here, the alpha-tocopherol protein is shown as just as an example. That was our collaboration with people from Canada, which we published in DCMB. And what this method our method can determine which residues will be penetrating to the axial chain region and how deep. And also we can calculate free energy of binding, transfer energy changes during association of these proteins with lipid bilayer. And we can compare these calculated values with experimental values for a number of different peripheral proteins. Not only for tocopherol, but for a number of others. So it was published before, actually. And there is a sufficiently good correlation, but it depends on computational models, which try to improve, and we actually improve this consistency in the second version of ppm probe. Or we can predict if we, for example, substitute this phenylalanine by some other residues like aspartic acid, how strongly it will affect the binding. And basically, these resources, the only existing resource, our ppm web server, which allows modeling peripheral rather than transmembrane proteins in the bilayer, freely available on the internet. So our PM database, we have put the results of calculations. It includes 10,000 PDB entries, and these proteins also classified to different classes, superfamilies and families. And there are maybe around 1,000 of different families and 500 superfamilies. It also provide topology and intracellular localization, so you can actually browse proteins based on localization, species, families, and so on and so on. And if you look at the content, you can see that transmembrane proteins, alpha helical and beta viral, they constitute less than half of all proteins in the database. All others are peptides, which also associate with lipid bilayers, amphipathic and transmembrane peptides, and present in the PDB. And others are water-soluble proteins, peripheral or monotopic. Some of them can associate even permanently. But there are many of them, and this data cannot be found in any other resources or databases on the internet. So, and also included is ppm web server.
So how can you differentiate protein facing outside and inside the cell?
Yeah.
So your energy function can tell how much a protein binds to a lipid, how deep it is in the membrane.
Yes.
But it cannot tell whether it faces inside or outside the cell.
You mean can we define topology? In this database, we take topology from experimental sources. We don't do like an initial prediction of topology. No, there is like positive inside rule, for example. Yeah, I'm talking about topology.
I'm not sure what you mean by topology, but say whether
how it is oriented to inside or outside.
Yeah.
So there are two different orientations. It can be oriented this way or another way, for
Yes.
example. Yes, exactly. This is called transmembrane topology. So that information, it is important to know topology because the intracellular and extracellular parts are different. So basically, usually topology is well known for proteins in the PDB, and you can look at the publication by crystallographers. It is there. So we simply take topology from these publications. So we don't try to predict it here.
Okay. So the simple answer is you don't.
Yeah, you simply take this way or this way, whatever is indicated in the corresponding publication about the protein. So this is PM is very widely used. It had like almost 1 million unique visitors over the many years, and we usually have like 1,000 visitors per month. And it was cited in the literature in publications more than 1,000 times. So it is widely used, and it is also integrated in other bioinformatics resources, even in the protein database. So there are actually links in the protein database which are connected to our PM database.
Now, do you have a breakdown? How many are pharma versus pharmaceutical companies?
We didn't analyze it, but
That would be good to know. You know,
but some of them are from pharmacy.
I bet they are.
I had a lot of correspondence over the email with different people about it, and maybe a significant part of them was from the pharmacy, but most of them probably academics, yes, or students or researchers. And this is kind of like our current deployment diagram, how our resources are allocated. Because initially everything was located at the University of Michigan, and we used PHP and MySQL. But recently we decided to increase a little bit sustainability, and right now we only use Genomewide Research Cloud at the university for calculation. This is very, very useful thing actually, and it's free of charge and pretty significant computational resource.
Yeah.
For all our web servers.
This is really a very good resource, and it would be available to us. It's densely it has a dense computing stack. It's very high speed, good data I/O, and it's free and available to us. And some of the algorithms that we run, crash, flux, this kind of thing, they work really nicely on Yottabyte. So it's something to check out.
So this is a very nice resource and helps to save money. But all other our resources right now located, we try as an experiment to come to locate them in another place. So we use like Google Cloud right now for data storage, and we use Heroku, Firebase, and we also use Bitbucket for this.
Wow.
So we try to do some things because we work with computer science students, and they like trying different things, something to change. Because right now the way they do it, they were taught to do it, is different from something that we did like 10 years ago, 15 years ago. Before it was only PHP, PHP and MySQL buckets. It's good, but okay. So our PPM web server allow to calculate spatial arrangement in memories of peptides or proteins using their three-dimensional structure as input. The calculations usually fast and produces coordinates of the protein structure with membrane boundaries, and it works better than in terms of precision than some other tools, which can be used only for transmembrane proteins, because this is the only tool that can be used right now for peripheral proteins. So that was like a first part of my talk today, and the second part is about single screening by topic membrane proteins and tools which we created for that purpose. And basically, single spinning transmembrane proteins, they have only one transmembrane alpha helix, but they have a lot of water-soluble domains, frequently on the both sides of the membrane, and they are highly, highly diverse. And if you look at the human membrane proteome, for example, almost half of human membrane proteome are bitopic proteins. So they are very numerous. There are more than 2,500 of them in the human proteome, and they belong to receptors. They provide interactions between cells involved in adhesion. Some of them are enzymes, transporters. They are involved in membrane trafficking, like intracellular membrane trafficking, and so on and so on. And they belong to many well-known famous families like receptor tyrosine kinases, integrins, cadherins, and so on and so on. And all of them have different structures. So this is a very important class of proteins. More than 600 of them in human proteome of this 2000 are involved in development of different diseases, from cancer to diabetes or viral infections or whatever. And some of them are involved in the signal transduction, like these growth factor receptors, from the outside to inside the cell. And they usually have pretty big water-soluble domains on the both sides. And they frequently form dimers, homodimers, or heterodimers. And during the binding of the ligand, they change. There is a significant conformational changes in this part, water-soluble, and the extracellular part. And then the mutual packet of these two alpha helices changes. And the signal transduced inside the cell, where the conformation of receiver and activator domains became different again and asymmetric. And then they interact with some other proteins. So, but basically what we are interested in here is only this part, transmembrane part. So we would like to model three-dimensional structure of these homo or heterodimers. And it is important to model them in different conformational states, because they change conformational state during signal transduction. And this can be done actually for many thousand these different bitopic proteins in the entire proteome. So what we do here, basically we can take amino acid sequence, not a structure, from Uniprot or from other sources, run it from our HealthMap and 10doc web servers or programs which we use, and they do modeling of individual alpha helices or alpha helical dimers. And the result of this modeling we put into our membranome database, which includes three-dimensional models of dimers and monomers and so on. So the first part of this software is our program HealthMap, which calculates structure and stability of alpha helices in complexes with membranes, which could be at the surface, transmembrane, and also right now I'm working with these beta sheet models. And in that case, the coil in aqueous solution is considered as a reference state. And we consider like 2 steps of the process, like first formation of alpha helix in water, and then a transfer of the alpha helix from water to the membrane interior. So the energy of binding consists from 2 parts, folding and transfer. And to calculate this first transfer part, we use our thermodynamic model of alpha helix and beta sheet formation in membranes. It was published some time ago. And in that case, we are interested to calculate this energy difference between coil in aqueous solution and the folded structure of alpha helix or beta sheet. And this energy expression includes some terms taken from mutagenesis studies like alpha helical propensities or beta sheet propensities, and some other terms which can be theoretically evaluated or empirically taken from various sources. And the second term, transfer, is exactly the same as for APM program, which we talked about before. So, and of course, we're using atom representation of peptides and the same anisotropic solvent model. But this is for just individual alpha helices, for example. But then we need to calculate structure of the dimers. For example, gamma or heterodimers within the lipid environment, when the both alpha helices are situated within membrane and then they interact and associate. And in that case, we developed this 10-dot complicated 10-dot program, which includes uses amino acid sequence as input, then HealthMap, the previous program, generate models of linear alpha helices, and then different initial structures of the dimer are generated using some kind of template-driven sampling or template-driven threading procedure, and it includes local energy minimization in space of torsion angles using my program Conform NMR, which I developed many years ago. And then finally, it calculates the free energy of association, and this is the most important part, because there are different actually modes of helix-helix association here. And to identify the native-like structure, you need to be able to separate it from other alternative, possibly misfolded structure. And also, it is possible that there is no any association at all. Maybe these 2 alpha helices are completely separated and don't form any dimer because it is not energetically favorable. That may happen as well. So you need to use this free energy of association, which can be compared with corresponding experimental values. And this energy of association includes different contributions, such as changes in conformational entropy of side chains during binding, salvation or transfer energy within the membrane, interactions of helix dipoles, hydrogen bonds and van der Waals interactions, basically packaged between 2 alpha helices. But the parameters of energy functions here are different from the standard molecular mechanics or molecular dynamics, and were derived from protein engineering data. So we verified this our 10-dot method, and there was 2 different ways to verify. First, we needed to compare experimental and calculated energies of helix-helix association, free energies, and they were measured for many published for many transmembrane gamma dimers in micelles and lipid bilayers. So there is a really good correlation, but we have a little bit overestimated value of energy using our computation.
So you do microcalorimetry, you
Yeah,
actually
yeah,
make measurements, thermodynamic measurements?
The experimental data are already published in the literature, so we took everything from the literature. We don't do any experiments. We do the computation and compare with published experimental data for the same dimers. And when we did this ab initio or template-derived docking, we calculated these structures with RMSD with experimental NMR-based models around 1.5 to 3 arcs. So this is more or less sufficiently good precision, and we also did verification using not only NMR data, but many such peptides, bitopic proteins, were studied using site-directed mutagenesis, scanning mutagenesis, and ToxSCAR. There are more than 30 of them were published, and we also did comparison with these data. And then we decided to use it for genome-wide modeling and analysis of gamma dimers, only gamma dimers, not heterodimers. And what we did here, we took like 6,000 proteins from different species, including human, Arabidopsis, Dictyostelium, yeast, E. coli, and so on. And we finally, after we selected around 2,000 most stable transmembrane gamma dimers and included them in membranome for analysis. And what happens? There are some interesting trends. For example, we found that transmembrane gamma dimers are more abundant in higher eukaryotes. And mostly, like, for example, for human, you have a significant larger number than for yeast or for Arabidopsis. And this is not so surprising because these proteins and the dimers are involved in cell-cell interactions. So this is like multicellular organism versus unicellular organism. And most of them are receptors. You can look at the distributions by species, by functions, or by different types of biological membranes. And what happens, most of these dimers are situated in the plasma membrane in the higher eukaryotes. And most of them are receptors. But there are also many enzymes, and they are not necessarily situated in the plasma membrane, but in intracellular membranes. And another interesting question, there are two different spatial organizations for these gamma dimers. Some of them can be defined as right-handed with negative crossing angle, and other like left-handed with positive crossing angle. And these with right-handed are mostly, I mean, they are less stable, they are flexible, and they usually present in receptors, and therefore they can be more frequently found in plasma membrane. But in Golgi, in AR, mostly we can find dimers formed by enzymes with more stable left-handed dimers. So there are some interesting trends around here about. No one did this analysis of gamma dimers before for any organisms. And the results are currently available in our membranome database. 6 species, 6,000 proteins, 2,000 dimers. They are also classified to more than 1,000 of families, and they can be easily browsed here. The database also includes some genetic variants or mutations in transmembrane alpha helices, and these mutations are frequently related to diseases, like cancer, especially in case of cancer, and many other hereditary diseases. And we also provide complexes, access to, like link to complexes in the protein data bank for the responding proteins and protein-protein interactions. But this is all the interactions between two different bitopic proteins, like only proteins which can be involved in interactions of transmembrane alpha helices. And also topology, intracellular localization, and so on. So this is pretty significant resource with a lot of data. And two web servers, one of them is HealthMap for modeling of alpha helices in different conditions, and can be used for peptides in water, micelles, lipid bilayer, for proteins, and then dog web server, which can be used for modeling transmembrane alpha helical dimers, and it produces different models of the dimers, and the models can be easily visualized using GNMO. So when we do this visualization, what happens? Actually, there could be several different models of the helix-helix packing using GNMO, this like helix boundaries, and you actually user can click at different structures, and the different structures will be shown in this output. And it will also residues involved in the interfaces will be shown in the output and their energies. So it's, in my view, it's like pretty cool actually. And third part of my talk today is about permeability of molecules through the membranes. And here there could be permeability of small molecules like drugs and permeability of peptides. And for small molecules, we made it like two separate programs and two independent web tools. Because in that case, you use a single amino acid sequence, and here you need to just take the structure of the three-dimensional coordinate of the small molecule drug from PubChem or some other resource. And they go through our Perm web server to calculate the, okay, I'll tell you about a little bit more. And the results are deposited in the database, but it's important this is not only permeability coefficient, but a translocation pathway, the orientations. Because when you move something across the lipid layer, it rotates and occupies different orientations, trying to minimize energy in every point of this during this movement. So, what we do in this method? We move the molecules across the lipid bilayer, and at each point of this pathway, we do optimal rotational orientation of the molecule. It rotates to expose to water hydrophilic parts in each point and bury non-polar parts of the molecule. Then we draw this energy curve. The energy curve depends on polarity of this compound, of course, and it can be very low barrier or no barrier, essentially no barrier for hydrophobic compounds, or it can be significant barrier for polar compounds. And then we integrate this curve to calculate so-called permeability coefficients, which show how fast the molecule will move across the lipid bilayer. And those permeability coefficients were measured experimentally for many different compounds in different experimental systems and can be compared with the results of our calculations here. So first we did comparison with black lipid membranes. And of course this is the most relevant system, because what we do in these calculations, we basically consider something like DOPC and see what would be the permeability rate for this DOPC. And BLM is something similar. It's, although it can be actually another model bilayer. And we did it for ionized and unionized compounds. And there is a pretty good consistency here with tilt-around unit and significant correlation. But the situation is a little bit more difficult for experimental data about permeability through blood-brain barrier. In that case, of course, there could be passive diffusion, but at the same time it could be also some transporters involved, or it can be some efflux involved. So it could be highly hydrophobic compounds that go to the cell, cross the cell, and then go back by exporting. But even in that case, there is a sufficiently good correlation between our calculations and the results of permeability through BBB. And it is possible to at least approximately distinguish compounds which penetrate or don't penetrate to the central nervous system. So it can be used as a predictive tool too, but with less successfully than in case of a real physical system like just BLM, where you have only passive diffusion without any possibility of additional active transfer or facilitated diffusion. And we created also this web server and database, which allow calculating permeability coefficients for small molecules. And there are different options for optimization here. It also calculates binding affinities of small molecule to the lipid bilayer, in addition to permeability rates. And we also created Perm database, which collect data for about 500 most well-studied drugs and the drug-like molecules. Well-studied meaning permeability coefficients were experimentally studied, which is also accessible in our websites. And this web server also provides a visualization of results of calculations. First, it provides the energy curve, how energy of the molecule changes when it moves across the lipid bilayer, and also provides pictures using the same GLMO tool, when you can click at different positions here and it shows molecule in different positions as it moves across the lipid bilayer. And we can even show kind of moving, how the molecule moves across the bilayer. So it rotates in such a way to expose polar group and bury hydrophobic groups, basically. This is ziprasidone is used as an example of a drug molecule. Okay. So okay. But again, this is a kind of free energy profile of transfer. And it's exactly the same model as for proteins and peptides, because this is like general physical model that can be used for proteins, peptides, small molecules, whatever. And finally, we are working right now to develop the cell-PM method for peptides. But it is currently under development yet, although the website is already available. And in that case, you can use as input amino acid sequence and look at the prediction of peptide ability to cross the lipid bilayer. Basically via passive diffusion. And again, in that case, you can try to look at the visualization. So this is positively charged residues are marked by blue, and they are trying to stay out of the membrane as it moves, as compound moves.
So in reality, you know, when you, in real life,
Mm-hmm.
you know, these are working through very crowded regions. It's
Yes,
not
this
just
is
this thing. I mean, you know, it's, you know, there's a lot of crowding in there. There's, you know, so I wonder how that would affect things.
No, basically you have lipids, and if this is fluid bilayer, this peptide or small molecule mostly supposed to interact with lipids. But of course, if you also have proteins,
Right.
it may interact with the protein surface.
Of course.
Bind to protein surface of transfer, and this may affect
That's my point.
transfer
Yeah.
rate. Yes. If you have just a transporter or channel, then of course it will be a completely different story.
Yeah.
But even with the surface, it may affect concentration of water, for example.
Yeah.
So this is the kind of approximation. So basically, this is the kind of reason. If you look at these curves, for example, for comparison with experimental permeability coefficients, but for black lipid bilayer, you don't have any proteins. But in
Right.
BBB,
For
you
that, it would
definitely
be
have.
obvious, but you know, in a real life situation,
but
it could
for-
be really quite different.
For blood-brain barrier, for example, you definitely have a lot of proteins. Yeah, and this is maybe one of the reasons for discrepancies, or the consistency is not so good as it might be. It is worse than for BLM, for example, definitely.
Right. Well, it might be possible to make a model. you
Yeah,
know.
so this is like, basically this is like a baseline. What
Yeah.
would happen if we don't have any active transfer? But let's compare what happens in reality. And then we have some discrepancies which can be explained by efflux or active transfer within the cell, for example. Yes. So you do need to consider this just as one of tools and one of possibilities. Yes, as a baseline.
So what contributes to the asymmetry of the energy profile in this case?
This is
Question.
yeah, I can explain. So there are different ways. We would like to calculate the energy barrier. For example, if we do rotational optimization without energy, just find the optimal rotational position in each point across the lipid bilayer. Then what happens? You take some molecule which is asymmetric, put it in the middle membrane, then find optimal orientation, which is basically flip-flop, and you neglect this energy of flip-flop. This leads to underestimated value of barrier. That's why here I'm using so-called, instead of doing just that, this is still one of possible options, and this exists in the circuit. But here I'm using so-called drag method, when you basically take the molecule and drag it through some point through the lipid bilayer. And when you drag it, you calculate energy in each position. And when you do that, you obtain asymmetric energy curve. Because what happens, you drag in some position, so it's still somewhere, but then it suddenly rotates. Just without any additional barrier, but just local energy minimization. In that case, you obtain more precise estimate of the actual energy barrier of the actual process, but it becomes asymmetric. So this is just two different options for optimization. It's not that simple, unfortunately. But there's a well-known problem. If you have like some kind of pass, energy pass, some complicated energy surface, and you move something, how to determine the value of like lowest point of the pass when we pass from one value to another value. And that is what happens here. This is not just local energy optimization when you just trying to find local or global energy minimum, but finding the pass. And this is can we do this? Yeah. Okay.
So then it's very dependent on the initial orientation, right?
Not really, because I allow it to rotate in the, initially I allow it to rotate freely in water. It rotates freely, but then it adopt some orientation and moves, but, okay. Actually, you can start from different initial orientation and see how it affects the results. It doesn't affect much. Because what happens is, but the form, shape of this curve may actually change, depending on the initial orientation, yes. But the results, no, basically what you have to do is a little more complicated, because you have we have a number of different conformers. So for example, for peptides, I'm trying to do local, like optimize, especially for peptide, optimize side chain conformers in each position. So I don't consider any energy barrier between different side chain conformers. We consider them to go freely from one orientation to another. This is, of course, approximation again. So, yeah. I'm trying to do, just don't make it too complicated, because we need to have a relatively fast procedure to be able to use it for web servers like that. We are not trying to do the molecular dynamics here. It should be a lot more complicated.
If you don't do ND simulation, how do you sample the information?
In that case, for example, for small molecules, the user is allowed to include like a set of different conformers. Basically, you can run through any kind of software and then generate a set of possible conformers like model 1, model 2, model 3 in PDB files, and then include them in the input file. If you do that, the program chooses the optimal conformer in different points of the transmembrane trajectory, but does not consider anything else. It considers each conformer as a rigid body. It's like a set of different rigid bodies. So again, this is like crude approximation. So it does consider different conformers, but do it in a very approximate way. And the value of the size of the initial sample can be different. You can include like 3 conformers, like 1,000. But if it's 1,000, then it will be slow. Yes. When we did it, we used some software. We did like cluster analysis, for example. Cluster analysis to different clusters representing different parts of conformational space. And you take just one representative for each cluster, and then you go run this set of conformers through the membrane. This is kind of better than just, no, basically to include conformational flexibility is important. Because, for example, rotation of side chains here are important, and for example, if you have something highly flexible organic molecule, you need to take into account the possibility it can be like that or like that. So you must include several conformers, but they must be sufficiently represented. How fine graining, maybe not for very fine grain, maybe very crude approximation, but again, it's kind of a trade between speed and accuracy.
Do you offer the option to generate those conformers for the user if they don't have the
Not really, not in these two, because there are some other tools existing for generating conformers for molecules. So I assume that user will generate, no, actually even PubChem provides a number of different conformers. So you're supposed to generate them using other tools, and then use it here as an input. But for peptides, I'm still working on it. It's not quite ready. The server is working, but the underlying program I'm still unpublished, I would say that. But for small molecules, Perm is practically ready. So, and finally, I would say about our future directions, because we have like big plans. One of, and we submitted several grant proposals, are planning to submit several grant proposals right now, and depending on what will be funded, we will may do different things. One of them is development of method for modeling transmembrane heterodimers rather than homodimers, or modeling of, in this would allow us to consider, to analyze transmembrane interaction networks of the topic proteins, because we don't know exactly, we know about interactions proteins A and B, but we don't know if they interact with water-soluble domains or transmembrane domains. And we can try to answer this question by using this software. And we can try to predict influence of passenger or driver cancer mutations in transmembrane domains of epitopic proteins. Or we can develop tools for modeling complexes between single alpha helix and multi-pass membrane proteins with known three-dimensional structure. Or we can look at the folding pathways and evolution of structures of transmembrane protein or multi-helical transmembrane proteins, because we can calculate, for example, stability of alpha hairpins. Like if you have two alpha helices, you can tell if it will be stable association in the protein structure or unstable association. If it's stable alpha hairpins, like in certain cytochrome B, for example, then it may be like initiating nucleation, which initiates folding of this polytopic transmembrane protein, for example. Well, just a general idea. And finally, I would like to make some acknowledgements. Right now, we have a little bit of fun remaining from the second age. And of course, I would not be able to accomplish all this work myself. And I would like to thanks a lot to Irina Pogozheva, who is here. And she is my constant, most important collaborator during all these years. Without her, it would not be possible to do at all. I would like to thank Andrei Lomize, who is my son. Actually, and he was a student here at the University of Michigan, and he developed first version of OPM and membranome databases. And Henry Mosberg from the College of Pharmacy, professor, who was very supportive at the initial stages. Yeah,
I remember him.
And Alan Byron is the system administrator who helped us with the setting up.
You worked for me years ago, 25 years ago.
Maybe you remember even.
Yeah, I do remember.
And also, we had a number of undergraduate computer science students who wanted to train using our resources and improve their skills, but at the same time they developed these web servers, wonderful web servers, and help with development of databases and so on. So it's kind of interesting playground for computer science students, I think. Thank you for attention.
Thank you. Okay, any questions?
So in your HealthMap, how do you sample the conformations? HealthMap need to hold the protein. How do you sample the conformation?
HealthMap it is for modeling of alpha helices, or for example, individual alpha helices. So this computational model is estimate free energy difference between coil in water.
Yeah, I understand the energy part.
How do I consider conformations?
Yeah, how you generate different conformations.
Basically, what I did is like calculations. If you have an alpha helix, this is a kind of standard geometrical structure, and you have different side chains in different positions. So what you can do, you can actually tabulate different types of interactions which may exist in, for example, for side chain situation in I+4, I+3, and so on position. So there are different parts there. There is a contribution from backbone, contribution of side chain, main chain interaction.
Energy calculation is one part.
I don't calculate it from the first principles.
Yeah, even your energy calculation is correct. You need to move your conformations from the initial structure to the final folding helix. So the question is, how do you move your conformations? Do you use empty simulation?
No,
or
I am using my own software which generate different sidechain conformers. There is a library of different sidechain conformers. For example, in alpha helix beta sheets like backbone dependence library of sidechain conformers. And then when you generate them, you can do local energy minimization in space of torsion angles using certain force fields, like for example ECPP or modified ECPP, which I am using right now. And then you can calculate conformational energy or this free energy, which is a different quantity. So I do conformational search using some libraries of conformers, for example, and I do local energy minimization in space of torsion angles. For example, for the this is mostly relevant to TENDO.
So you enumerate the conformations?
Yes, I enumerate different sidechain conformers. Yeah.
Okay, so
and also when I do template driven sampling, I actually do a library of sidechain conformers that are allowed in certain position of this template, because that allows to speed up significantly the calculations.
Okay, any more questions?
So I don't use MD because if I use MD, it will take like 100 times longer, or maybe many thousand times longer than,
Oh my God.
than here. Yeah.
Your algorithms do seem to run plenty fast. Do you spend much time optimizing them, or do they run in parallel at all?
Actually, this resource allows yottabyte cloud, allows parallelization.
You
Right
take
now,
advantage
I am planning to do some parallelization of the code, but I didn't do it yet. So basically, just sitting on a single processor. But at most, it usually takes maybe half an hour for the biggest structures. Like if you want to position in memory photosynthetic complex, like photosystem, huge protein complex. It takes maybe almost an hour. But for small, it's much less. But if you use it through email, you just receive the reply by email with the results, and then you have everything. But it's sufficiently fast.
Yeah.
Okay. Andrei and Marina, thanks very, very much.
Thank you.
Yeah, very nice work.
Thank you.
Yeah, we enjoyed having you. Thank you. Okay.